Greetings to all. My name is Nuratika Binti Ishaq. I would like to present my topic regarding the nervous system and behavior of weakly electric fishes. Before go any further, I would like to explain a little bit about Mormyriformes. Mormyriformes is a superfamily of freshwater electric fishes. It is mostly found in Africa. These fish are technically advantageous as experimental subjects in studying the neural and hormonal mechanism underlying the sexual dimorphism in communication behavior. Other than that, weakly electric fish generate a discharge typically less than 1 volt. These discharges are used for navigation, electrolocation, in conjunction with electroreceptors in their skin, and electrocommunication with other electric fish. Advantage of Mormyriform for behavior is that we can determine the electric produced by them regarding to their species. We are also able to understand their behavior when searching for food. As we all know, nervous systems are various in every organism. It can be seen to evolve from the most simplest organism, such as worms, to the most complex organisms, such as cephalopods. Thus, this topic made me wonder how nervous system in weakly electric fishes caught my attention. The thing that have been questioned in this research is how the brain region scaling evolved by using two hypotheses. The first hypothesis is known as mosaic hypothesis, which explains changes in the relative size of particular brain region. Concern hypothesis is where the brain evolved as a coordinated structure due to developmental constraints. Background of this research are mostly focused on the nervous system of weakly electric fishes and their brain evolution. Those information were learned to be able to understand the brain organization and structural delineation. Other than that, is to determine the brain volume and to determine whether a grade shift had occurred, besides to quantify shape variation using homologous landmarks. A few results and observations were obtained during the research. For example, enlargement of cerebellum occurred in both passive electrosensing and active electrolocation, including communication. In passive electrosensing, it occurs via ampullary electroreceptors evolved in osteoglossomorph fish, thus allowing for the detection of external bioelectric fields in the fish. While in active electrolocation and communication, the enlargement arose with the evolution of electric organs and tuberous electroreceptors in Mormyriformes. In addition, brain region providing an excellent system to test for mosaic evolution. Other than that, there are mosaic shifts that occur in brain region size. For example, in cerebellum and hindbrain, referring to the graph, Mormyriformes can be seen to have larger y-intercept compared to the outgroup species. However, outgroup species had larger y-intercept in telecephalon, OB, and OT. Last but not least, there is no significant difference in y-intercept for ROB. Brain also experience shift in their shape. According to the graph, shape variation along PC1 describe morphological changes in the cerebellum that occur in positive and negative direction. In positive direction, the cerebellum located in the posterior dorsal position relative to the ROB, while cerebellum will expanded in every direction, leading to a more globular overall brain shape in negative direction. Data in PC1 supported the concerted hypothesis mentioned in the early presentation and demonstrate that most variation in brain region size is highly correlated with total brain size. PC2 explains more in the variation. It demonstrates the dramatic shape change that occurs over the same phylogenetic timescales. This further emphasizes the dramatic brain region changes that occurred with the evolution of Mormyriformes. In addition, PC2 illustrates mosaic shift in brain region that separated Mormyriformes from outgroup species. Next, I will finish my presentation with discussion. There are limitations in this research study where the active electrosensing evolved only once within the osteoglossomorph. Thus, it is impossible to determine the extent where mosaic shifts are related to the evolution of active electrosensing. Other than that, brain region size that varies were explained by the mosaic shift that occurred. In this experiment, both hindbrain ELL, which is the furthest posterior point of cerebellum, and cerebellum, hold an important role in sensory motor integration, which are fundamental to active electrosensing. Mosaic enlargement are managed by the evolution of active electrosensing. An enlarged cerebellum may have been driven by constraint due to a late developmental plane shared with ELL. Telencephalon responsible to receive electrosensory input. Thus, the size of telencephalon may decrease relative to the entire brain size due to the decreases reliance on visual and olfactory processing. However, no shift occur in ROB due to limitations inherent in combining regions. Next, a study of brain development and neurogenesis indicated several neurogenesis zones in the CB that persisted throughout life. The extensive adult neurogenesis caused mosaic change more easily to occur. For brain organization in non-mormyroid, horizontal plane ran from the point of the TEL was furthest ventral in a straight plane back to the furthest dorsal part of spinal cord, while mormyroid horizontal plane ran from the point of the TEL was furthest dorsal in a straight plane back to the furthest dorsal bulge of the hindbrain. In hindbrain, CB could engulf HB both dorsally and ventrally. In non-mormyroid species, CB was a small ellipsoid at the farthest dorsal posterior end of the brain, while in mormyroid, CB was a helmet-shaped area that covered most of the brain dorsal area. Last but not least, the large variation in the size and shape of ROB region across the osteoglossomorph are due to the expansion of the cerebellum pushing the midbrain further ventral. By ending this discussion, I would like to end this presentation, and thank you to everyone.